In this lecture, I will introduce paradigm shifting concept and approach using genomic information, novel design, and advanced statistical tool. With this novel approach, I will show how to tackle problems that is also problem in your own research. So you can estimate heritability based on genomic information. You can estimate genome-wide pleiotropy, in other words, genetic correlation based on genome-wide information. You can estimate genome-wide gene by environmental interaction. You can also reveal latent genetic mechanism. So first, algorithm for linear mixed model. This is known as residual maximum likelihood. It's called RAMR. So in the last lecture, I show grid search and Newton-Raphson method can be used to estimate heritability. In addition to this algorithm, there are a number of different algorithms: Fisher scoring method, expectation maximization algorithm, and derivative-free RAMR, and average information algorithm. So this average information algorithm is probably the most efficient approach to solve linear mixed model. So this average information algorithm take average of Hessian, that is Newton-Raphson approach, and Fisher's information matrix. So this equation, that is a parameter in the next iteration, is a parameter in the current iteration subtracted for Hessian matrix, inverse times first derivatives. For the Fisher method, so this H matrix is replaced with Fisher scoring information matrix. Then this average information take average of this H and this Fisher matrix. So H matrix is the second derivatives of the likelihood. The likelihood is I introduced in the last lecture, right? That's sum of the product of probability density function and take a logarithm. Then Fisher scoring method is excluding actually that take only this trace term. Then average information is average of this H and F. Then the previous study shows this average information algorithm is computationally much faster compared to H approach or F approach. Average information algorithm was originally based on mixed model equation that is developed by Gilmore 1995. So mixed model equation looks like this, that include fixed term and random effects term, and the solving this equation. I'm not going to explain much detail. I'd like to just introduce the concept comparing between two different algorithms and their application. So this AI algorithm, average information algorithm, can be also based on V matrix, that is phenotypic variance covariance matrix. So this is directly from V matrix. So this algorithm has been named as a direct AI RAMR, and this is the original AI RAMR based on mixed model equation. Okay, so these two different approach has actually different computational performance depending on different situation. So for example, for the dense genomic relationship matrix, if there is no zero elements in genetic covariance structure, or there are many multiple random effects, in that case, direct AI RAMR that is for this, is much faster compared to AI RML based on mixed model equation, that is this. That's because uh, if random effects is added into the equation, then this dimension, as you can see, is increased, right? Therefore, computational efficiency is rapidly decreased. However, the same random effects added in the V matrix that for the direct AI RMR, this dimension of V never changed. Therefore, computational uh, demanding remain the same, whether using single random effects or whether using multiple random effects. Therefore, this direct AI RMR is much advantage of mixed model equation based AI RMR. So this direct AI RMR has been implemented in the GCTA and Multiblob and MTG2. These software widely used in human genetics. So you can download this software from their website. So one of the software I will show where you can download at the end of this lecture. So get back to the linear mixed model. So this is the number of papers published in agriculture and biological science that from Scopus and from 2000 to 2014. And red bar is the number of papers from linear mixed model. And blue bar is from linear model. And this green dot is a ratio using ln m over lm. So as you can see, in 2000, the number of papers for LMM is relatively smaller and ratio is only around 15%. But over the 10 years, 2014, it is rapidly increased up to the 40% in this field. And this is the field of medicine and is very similar pattern. So in 2000, the ratio is around 7%. Then over the 10 years, it is rapidly increased up to 20%. So this shows linear mixed model is emerging tool to tackle problem in biological science or medicine. Linear mixed model is a function of genetic covariance structure. So A is genetic covariance structure or relationship matrix that can be based on pedigree or based on genomic information. So this A I explained in the last lecture, and I will explain again briefly. Using the whiteboard. So if you have 1 to n individuals, then also you have phenotypes y1 to yn. Then this record or from based on this individual, there is a relationship, genetic relationship among this individual. Then you can get one matrix that is called A matrix, that is n by n. Then you can draw the each column and row, something like this. Then the diagonal is 1 because they are same individuals. First individual, second individual, first individual, second individual. Then if the first individual and second individual has they are full shifts, then you can specify as 0.5 here, and so on. So this is what does A look like, A matrix look like. Okay. Yeah. Then Traditionally, this A matrix is based on pedigree information. So full SIP is 0.5 and MZ twin is 1 or, and so on. But now the genome information is available, so this A matrix can be constructed based on genomic information. So this is a kind of paradigm shifting. Before going detail, I'd like to show briefly how to construct genomic relationship matrix. So this A, genomic relationship matrix, is a function of a product of genotypes from pair of individuals. So 2p is the mean of allele frequency. Then this x is genotype for individual I, this for individual j. Then their SNP genotype coefficient is centered, then scaled by the variance of SNP genotypes. Then also this S is a scale parameter. So I'm not going to too much detail. Then if you are interested, then please have a look this paper, 3 papers. Then it would be great if you like discuss about this with me. Then please contact me anytime. I'm really happy to discuss about this. So why this genomic relationship matrix is a paradigm shifting? That's because it can link between unrelated individuals through genome-wide SNP similarity. So previously, based on pedigree, there is no way to link between these two unrelated individuals, right? But now it is possible to construct the genome relationship matrix based on genomic information between any pair of individuals that come from the population. Therefore, now it doesn't need pedigree or twin design anymore. Is all design-free experiment is possible, and heritability or genetic correlation can be estimated based on these genome-wide SNPs. Also, this genome relation matrix can be used to link between two independent groups. For example, these groups and these groups. Previously, there is no way to link between this group if they are basically unrelated. But now, as long as they genotype for the same set of SNPs, then you can work out based on the SNP similarity, genotype similarity. Therefore, you can construct the relationship between any independent groups. So, because of this paradigm shifting concept, now it is possible to estimate genetic correlation between any pair of groups. For example, IQ in general population that compare with people with schizophrenia. So this kind of experiment never been possible before, but now it is possible based on genomic information. Also, this is in the context of livestock genetics or animal genetics. Probably some of you are interested in pursuing your research in this field as well. Therefore, I include this one. So previously, based on pedigree or traditional design, it never been possible to link between different breeds. For example, Hanu, this is Korean cattle, and Angus, this is in popular breed in the America or Australia or any other countries. But now, based on SNP information, as long as they genotype on the same set of SNPs, then you can work out the similarity, SNP similarity or genotype similarity. then to get the genetic covariance structure between two different breeds, then it's possible to estimate genetic correlation between these two breeds. That never been possible before. Okay, so this is in the context of genotype by environmental study. So this novel concept and approach can be also applied to G by E study. So for example, if we want to test if there is a G by interaction when people born in winter or people born in summer. However, this never been possible because in traditional design, if you want to tackle problem in G by E context, you have to have repeated measures. For example, in this case, one have to have a multiple record for the when they born, but because of this is birth information, it's not possible for anybody have multiple repeated measures, right? Because only people or people has only one birth date, not multiple date. That's why such GBI study never been possible in traditional design. However, based on genome-wide SNP information, you can group people born in winter and people born in summer. Then, as long as they have a genotype on the same set of SNPs, then you can work out that genotype similarity between this group and this group, then estimate their genetic correlation and to test whether this group and this group has a heterogeneity. There is a well-established statistical theory that can be used to tackle in this problem. So I will show a few examples how this novel approach and concept has been applied to tackle real problem. So this is published in 2012. So this y-axis is heritability, and this x-axis is chromosome length, and this is chromosome 1 to chromosome 22 fitted in linear mixed model to estimate genetic variance or heritability. So you can see the estimate heritability for each chromosomes, and this is positively correlated with their chromosome length. So this shows that schizophrenia is a highly polygenic trait without no major genes. There is no deviate from the expectation. But what I like to point it out is with the novel approach and novel design, it is possible to partition each chromosome genetic variance or heritability across genome. However, this kind of analysis never been possible by using traditional design or traditional approach. So in this case, this is extension of a single variate linear mixed model to estimate genetic correlation between two traits. So in this study, there are 5 different psychiatric disorders: schizophrenia, bipolar, MDD, ADHD, and autism. So this study found that empirical evidence that genetic correlation between schizophrenia and bipolar is high, and schizophrenia and MDD is moderate, and schizophrenia and autism is low. Again, this kind of study never been possible in traditional approach or design. However, with genomic information and the linear mixed model, it is now possible to estimate the heritability and genetic correlation. In your own research, you need topics or title, you need to set up hypotheses and aims, and you need methodology. In order to test hypotheses, also you need data. So from the analysis, you will get the result, and you need to correctly interpret the result. Then also you want to validate your finding in independent dataset and conclusion. So this lecture and practical will help understand know why and know how, especially in methodology. For example, linear mixed model for complex trait analysis. So after this lecture and practical, then you will have knowledge how to use linear mixed model to estimate heritability or genetic correlation that are genetic parameters. Also, you can use software not only for research. Also this tool can be used in a wide range of industrial professions. For example, some statistical survey or some data analysis or some summary statistics needed. So in practical, you will use a linear mixed model to estimate genetic parameters, and you will use software MTG2. You can download from this website, and there are some example files in the this folder. Then you can use and follow the practical instructions. And it would be great if you give any feedback that can improve this lecture and practical. Thank you.